Hello, everyone. In this lecture, we are going to be studying respiratory physiology, the function of the respiratory system. Now, some of the concepts in this lecture might be tough to understand initially, but stick with me. We'll break it down and make it so you can understand some of these most complex concepts in anatomy and physiology. Ready? Let's go. So we can break our study of respiratory physiology into four main processes. Pulmonary ventilation, and this is just breathing. This is the movement of air into and out of our lungs. We can also talk about pulmonary gas exchange, and so that's where gases are going to be moved at the microscopic level between the lungs and the blood, and tissue gas exchange, where we're going to move gases between the tissues of the systemic circuit and the blood of the systemic circuit. And finally, gas transport, how we're actually going to make sure that gases get from the lungs through the blood to the tissues and back again. So let's get started talking in detail about each of these. So let's start off by talking about pulmonary ventilation. Again, this is the same thing as saying breathing, inspiration and expiration. And it's going to be based primarily on pressure gradients. So you might remember the term gradients all the way back from earlier units. Remember, a gradient is just where something has a different value on one side of a membrane than on the other. So for example, you've talked a lot about concentration gradients, where the concentration of molecules is greater on one side than it is the other side of the membrane, and thus things want to go from an area where they're more highly concentrated to an area where they're less concentrated. So that was the concept of diffusion and osmosis that we talked about. Well, a pressure gradient is a similar idea, except instead of concentration of molecules, we're talking about the pressure put on molecules. So just like with concentration gradients, in pressure gradients, gases want to move from an area where they have higher pressure to an area where the pressure is lower. And related to this is the relationship between pressure and the volume of the container that the molecules are in. So we're going to look at a law called Boyle's law. And Boyle's law says that pressure and volume are proportional to one another. In other words, when there's very little volume, when the volume is low, the pressure is going to be high, and vice versa. When the volume of the container is high, the pressure is going to be low. So let's take a look and see why that is. Let's check out this diagram right here. You see a container with a large volume, and the molecules represented by the red balls here are able to freely move by, and very rarely do they encounter each other. In other words, they have more space, so there's less pressure on them to be touching. They're able to move about. There's less pressure on those molecules because there's a large volume. In contrast, the container over here has very little volume. It's a small volume, and thus these molecules are all crowded together. They're bombarding each other. They're under higher pressure. So think of the movies where you've seen the walls that close in on people and they're stuck in this room. Well, as the walls come in, the pressure goes up. So same kind of idea, that instead of people, we're talking about molecules. As those walls close in, the volume gets smaller, the pressure on those molecules increases. So this is going to be a very important concept for all the aspects of respiratory physiology that we're going to be talking about. So when we're talking about pulmonary ventilation, we're going to specifically be talking about three different pressure gradients. So first of all, we're going to talk about the pressure of the atmosphere, atmospheric pressure. So the air that's out in the environment is under constant pressure from the force of gravity. And so this pressure applied to the molecules in the air is going to be at 760 millimeters of mercury. 760 millimeters of mercury is our atmospheric pressure. Now, moving on down into the respiratory system, we have the pressure that's applied to the air inside the alveoli of our lungs. And this is called intrapulmonary pressure. So the pressure inside the alveoli. So at rest, meaning between breaths, between inspiration and expiration, the pressure inside the alveoli, the intrapulmonary pressure, is going to be equal to the atmospheric pressure of 760 millimeters of mercury. Now, this of course can fluctuate during breathing, but overall the net effect is that eventually the pressure inside the alveoli will be equal to that of the atmospheric pressure, 760 millimeters of mercury. Now, recall that outside of the lungs is a cavity called the intrapleural cavity. The intrapleural pressure is going to be always 4 millimeters of mercury below the intrapulmonary and atmospheric pressure. Now, it too can fluctuate during breathing, but on average, at rest, so between inspiration and expiration, the pressure in the intrapleural cavity is going to be 756 millimeters of mercury, 4 millimeters of mercury below that in the alveoli. And this diagram will remind you of where we're talking about with the intrapleural cavity. So here are the lungs with the alveoli, and there's the thoracic cavity. The intrapleural cavity is where the lungs sit. Okay, so you just learned that during rest, i.e. between inspiration and expiration, our alveoli inside of our lungs have the same pressure as the pressure in the atmosphere, 760 millimeters of mercury. So they're equal. But now let's add in the variables of inspiration and expiration. So during inspiration, we're breathing air in, and the volume of our lungs is expanding. It's getting bigger. So recall from Boyle's law that the higher the volume, the lower the pressure. In other words, the bigger the volume, the more space there is for the molecules to move around. They're under less pressure. And so during inspiration, our intrapulmonary pressure becomes less than that of the atmosphere. It drops down to 758 millimeters of mercury, slightly less than the 760 of the atmosphere. And so that sets up a pressure gradient, right? The pressure inside the lungs in the alveoli is going to be less than the pressure in the atmosphere. And given that it's a pressure gradient, molecules like to move from an area of higher pressure to lower pressure. And so the air molecules will move from the environment, from the atmosphere, down the pressure gradient into the alveoli of the lungs. So inspiration causes air into the lungs because the pressure is greater in the environment than it is inside the lungs. Now, of course, it's the opposite for expiration. With expiration, we're talking about the intrapleural cavity, the area around the lungs. And so when we expire, your diaphragm moves up, and that decreases the volume of the thoracic cavity. Less volume, more pressure on those air molecules. And so the pressure of the intrapleural space becomes greater than the pressure in the atmosphere. Lower volume equals higher pressure. And so the pressure in the intrapleural space goes up to 762 millimeters of mercury versus the 760 in the environment. There's a pressure gradient. Molecules like to go from an area of greater pressure to lower pressure, and hence air molecules are going to move out of the intrapleural space, out of the lungs, and into the environment. Now, it's worth noting that your alveoli inside your lungs are always going to remain inflated. If they didn't, we'd have a problem. And so there's always going to be a slight pull on the lungs from the pleural cavity. So this is related to, for example, the parietal pleura, pulling on the visceral pleura, which pulls on the lungs. So that keeps the alveoli always inflated. So in the end, when you take the pressure gradient that's created through inspiration and you look at the pressure gradient that's created through expiration, the net value of pressure is going to be the same as it is in the environment. So in the net value of inspiration and expiration, the pressure through inspiration and expiration is going to equal the pressure of the atmosphere, 760 millimeters of mercury. Now let's really quickly review some of the concepts we just mentioned as they relate to the muscles of the respiratory system. So we were just saying that there's always a little bit of a pull on the lungs, and that helps to keep the alveoli open at all times. Well, part of this is thanks to our thoracic skeletal muscles that, as they contract, will pull on the lungs. Specifically, they're going to pull on the parietal pleura and the visceral pleura, and ultimately help to increase the volume of those lungs so that the pressure is less, so that the air can move. Also aiding this are going to be the external intercostal muscles, and of course, as we mentioned, the good old diaphragm here. So this diagram is going to show you what's happening with the diaphragm muscle upon inspiration and expiration. So you can see how it changes the volume. So when we breathe in, inspiration, that's going to cause the diaphragm to flatten out. And when that diaphragm flattens out, that's essentially increasing the volume of the thoracic cavity and therefore decreasing the pressure. Versus when we breathe out and exhale, the diaphragm's actually relaxing, but it relaxes into a dome shape. And so that dome shape takes up more space in the thoracic cavity than the flat shape did. So we're decreasing the volume in the thoracic cavity, hence increasing the pressure, and the air wants to move out. So just to restate what we just said, the dome shape is the relaxed shape of the diaphragm. That causes the volume of the thoracic cavity to decrease, hence the pressure in the thoracic cavity to increase, and in fact to increase above what it is in the atmosphere, setting up a pressure gradient. The air wants to move out. Versus when it's constricted and it's flat during inspiration, that increases the volume of the thoracic cavity and of the lungs, and thus decreases the pressure below what it is in the atmosphere. The air is able to go from the higher pressure in the environment to the lower pressure in the lungs. Air moves in. And it's worth noting that between breaths, that diaphragm's actually still going to stay in its flat state, its constricted state, so that the lungs don't collapse. Otherwise, if the diaphragm relaxed, then the lungs would collapse. So it's always going to be constricted between breaths so that the air in the lungs, there's always a little bit of air in the lungs keeping them from collapsing. So the muscles we've been studying thus far having to do with the respiratory system are geared towards normal breathing. So just normal inspiration and expiration that we don't really think about. But as you know, we can force inspiration and force expiration. We can take in more air than would be normal and expire more air than would be normal. And so when this happens, we're going to have to add in some other muscles. And so these are called accessory muscles of inspiration and expiration. So for forced inspiration, this means we take a big breath, right? That's forced inspiration. We're going to need the help of the external intercostal muscles again, plus your pectoralis minor muscle, which you remember is in the chest, the sternocleidomastoid muscle, which helps us to turn our head, and the scalene muscles. So if you recall your muscle anatomy, you'll remember these muscles. So they're going to help to greatly increase the thoracic cavity and the lungs and greatly decrease the pressure in the alveoli. So we're forcing it in with the help of these muscles. That's going to really drive down the pressure in the alveoli, and that air is going to come in a big way, right? Same thing with forced expiration, except in the opposite direction. So for forced expiration, we're going to really push that air out. We're going to need the help of the internal intercostal muscles plus the abdominal muscles. So that's our rectus abdominis muscle, our obliques, right? Your external and internal obliques and transverse obliques, and the quadratum lumborum. So some of those back muscles. So those guys working together are going to greatly decrease the volume of the thoracic cavity and lungs and greatly increase the pressure of the air in the alveoli, forcing that air out. Forced expiration. So this is really important because we're going to visit these concepts again when we talk about pulmonary volumes and capacities. The idea that you have regular inspiration and expiration breathing, and then you have the ability to have forced inspiration and forced expiration. So we've talked about just normal breathing, and we've talked about forced inspiration and forced expiration. But let's look a little bit closer at quiet or passive expiration. So when we're just breathing out some things normally, some air normally, we actually don't need to really engage muscles specifically. It's kind of a passive process. The diaphragm is just relaxing on its own, and those elastic fibers of the lungs are going to have recoil. So it's going to just return those lungs, the lung volume, back to normal without the help of added muscles. So passively, that volume is going to decrease. bringing up the pressure inside the lungs, and that air is going to move out. Now, there are non-respiratory movements as well, like you see Sleepy doing here, and that is things like yawns and sighs and sneezes and coughs. So these things, I remember as a kid them saying that they didn't know why we yawn, but good evidence is that yawning actually does play a role in respiration. So remember, we don't want those alveoli to ever collapse. We always want them to stay open, and when you're sleeping, they're more likely to collapse. So yawns when you get sleepy, when it's bedtime, that's going to help increase the amount of air in those alveoli, stimulate the release of something called surfactant that we'll be talking about later, but basically it's the coating around the alveoli, and all of that's going to help to keep those alveoli open at all times so they don't collapse. There are 3 factors affecting pulmonary ventilation. Number 1, airway resistance. Number 2, alveolar surface tension. And number 3, pulmonary surfactant. So let's talk about each of these, starting with the first one, airway resistance. Now, if you've studied the cardiovascular system, you might remember that blood vessels have the ability to constrict and therefore increase the resistance to blood flow, or to dilate, which opens things up, decreases the resistance, and blood can flow faster. Well, same principle applies to the respiratory system. So we have these tubes that conduct air in our respiratory system. And when you get to the smaller ones, especially like bronchioles, there's smooth muscle that creates the ability of those tubes to constrict or to dilate. And then recall also that there are muscles in the thoracic wall that are constantly pulling on the lungs, or the recoil of the elasticity of the lungs allows them to go backwards. And so this natural pulling and recoil that happens during normal breathing is going to affect the diameter of the tubes that conduct air. So, for example, during inspiration, recall that as we're inspiring, the volume of the lungs is expanding and they're getting pulled on by the thoracic wall. They're also pulling on those tubes, such as the bronchioles. And so very slightly, but nonetheless significant, that pull is going to create slightly more dilated vessels or slightly more dilated air conducting tubes. And it's going to pull them open, and thus air can travel faster through the tubes because there's now less resistance. The opposite is true of expiration. So recall during expiration, we're talking about recoil. Those lungs are recoiling, and thus that is kind of putting some relaxation on those conducting tubes, and they're in a sense constricting. The diameter is getting smaller. There's therefore more resistance, and the air is flowing through slower. Now, in addition to that just natural phenomenon that happens from the pooling and recoil during breathing, recall that bronchioles have smooth muscle around them. And that smooth muscle is going to lead to bronchodilation, the expansion of the tubes, or bronchoconstriction, the constriction of the airway openings. So when you have bronchodilation, the lumen is larger, and so there's less resistance. Air can travel faster. When there's bronchoconstriction, that lumen is smaller, and so there's more resistance. Air does not travel through as fast. Now, bronchodilation happens when we're under stress, or there's some emergency, or we're exercising. Recall that during those kinds of activities, you need more oxygen getting to your tissues. And so remember from the cardiovascular system that during stress, you might emit norepinephrine, which is the hormone, one of those fight or flight hormones that makes your heart go faster, but it also will cause bronchodilation, so causing those respiratory tubes to increase in diameter, lowering the resistance, and therefore allowing air to go through faster so we can get more oxygen to our tissues. During bronchoconstriction, there's increased resistance. Air is going to flow slower. This can happen, for example, during an inflammatory response, such as an asthma attack or an allergen. That can cause inflammatory mediators to be released, such as cytokines, which inflame everything and cause more mucus to be secreted. That mucus is going to decrease the resistance and slow down the airflow. This can also happen just because inhaled irritants cause parasympathetic neurons to be triggered. Or you can get an increase in resistance from things like tumors, which are blocking the flow of air. Either way, we're getting signals to those smooth muscles to constrict the tubes, and thus we are increasing resistance and slowing down the flow of air. So the first factor was airway resistance. Another factor affecting pulmonary ventilation is alveolar surface tension. So recall the structure of water molecules. Remember, water molecules look like Mickey Mouse, right? They have two hydrogen ears and an oxygen face. And recall that hydrogen ions on the ears maintain a partial positive charge, whereas the oxygen maintains a partial negative charge. This allows one water molecule to stick to another water molecule because of the hydrogen bonds that are formed between one of the hydrogen ion ears and the oxygen face of the next water molecule. And so these water molecules are all sticking together right along that gas-water boundary of the alveoli. And that is surface tension. So think of surface tension as the water film that forms. So remember when you fill a glass with water and you fill it a little bit above the top of the glass, you can still have water up there above the edge of the glass because of surface tension, or why water strider insects are able to float on the water. It's due to the surface tension. So surface tension at the gas-water boundary of your alveoli is not a good thing. That tension can actually cause the alveoli to collapse, and we don't want that. Remember, we want our alveoli open at all times. So thankfully, the type 2 alveolar cells of our alveoli secrete a special substance called pulmonary surfactant. Pulmonary surfactant looks like this. This would be one molecule of it. And you'll notice that it has a polar head. Polar likes polar, right? And water molecules are polar. So the polar head of the surfactant will actually bind near the water where the surface tension is and break those hydrogen bonds. That breaks up that surface tension and keeps those alveoli from collapsing, keeps them open. So thank goodness for pulmonary surfactant. And there are diseases of the respiratory system that happen because there's not enough surfactant being produced. And finally, the third factor affecting pulmonary ventilation is pulmonary compliance. This is basically how stretchy things are. So recall the word distensibility refers to the ability of something to stretch. And so the stretchier the thoracic wall is, the stretchier the lungs are, the more the effect on pulmonary ventilation there is. And so what makes something stretchy? Well, it can have to do with the elasticity of things like the lungs, but it also is affected by surface tension and the ability of the thoracic walls to stretch. So all those things together affect the distensibility, which is going to affect pulmonary ventilation. So that distensibility of the lungs in the thoracic cavity is what's known as pulmonary compliance. Okay, so now that we have the basics of pulmonary ventilation, we know how we breathe in and breathe out. Let's see if we can quantify it. So we're going to look at two things called pulmonary volumes and pulmonary capacities. So this will allow us to have a quantitative and graphical representation of how air moves in and out of our respiratory system. So let's first talk about normal breathing. So recall we talked about normal breaths versus forced inspiration, where we're forcing air into our lungs, and forced expiration, where we're forcing air out. So the normal breathing that we do when we're not being conscious about it is called tidal volume, or TV. So think of the tide goes up and down in an ocean. So this tidal volume is going to be the amount of air exchanged during normal breathing, and it's about half a liter or 500 milliliters. So let's go ahead and start building a graph that will show these pulmonary volumes. So we're going to go right in the middle and write these little waves that represent tidal volume, normal, inspiration, and expiration. And again, we said it's about 500 milliliters of air exchanged in normal inspiration and expiration. So we're going to start the graph at about 2,500 milliliters of air and go all the way up to about 3,000. So that's half a liter exchanged, and we draw it up and down. But as we said, we can sometimes force air into our lungs. And so this would be known as the inspiratory reserve volume, or IRV, the total amount of air exchanged by forcing it in after our normal breathing. So we're not going to include the tidal volume or normal breathing here. We're just going to look at the amount of air coming in on top of that. So that's our inspiratory reserve volume, and it can range from 2,100 to 3,300 milliliters of air inspired after our normal breathing. Different books say different numbers, as you'll see on the next graph, but in general, somewhere around there. And it should be noted that females tend to be lower on the end and males higher on the end of total air exchanged. Expiratory reserve volume is the amount of air expired after normal breathing. So on the graph, here's our normal breathing, our normal tidal volume, and under that is our expiratory reserve volume. And this is going to range from 700 to about 1,200 milliliters of air expired after normal breathing. And finally, you'll always have air left in the lungs after you've tried to blow it all out. And that's really important because if you didn't, our lungs would collapse. So we always keep a reserve, an amount of air in our lungs at all times. So this is called our residual volume, or RV. So it's down here on the graph. It's after your expiratory reserve volume. So this keeps our lungs inflated at all times. And if we were going to do a spirometer, as you see here, it's going to basically infer residual volume, but not measure it directly. What it is going to measure directly is your tidal volume, your inspiratory reserve volume, and your expiratory reserve volume. So it's basically measuring all this, but not the residual volume. Okay, so now that we've gone over pulmonary volume, let's now go over pulmonary capacity. Now, what is the difference there? Well, remember the volumes we talked about were for individual things that go on during breathing. So tidal volume, TV, that was our normal breathing, inspiration, expiration. IRV, or inspiratory reserve volume, was the amount of air exchanged, specifically through inspiration after tidal volume. And the expiratory reserve volume was the amount of air moved after tidal volume when you forcibly expire. And then we had residual volume, RV, which is the amount of air left in the lungs after you've forcibly expired. So we're going to now translate those into capacities, which are where you have two or more of those pulmonary volumes combined together. So let's start off with our inspiratory capacity, which is your normal inspiration breathing plus the amount of air moved from forcibly inspiring. So it would be like this, right? It's normal plus forced. And so that's your tidal volume plus your inspiratory reserve volume. So that is the total amount of air exchanged from inspiring. And looking on our graph here, so remember you had your tidal volume, you had your inspiratory reserve volume. We're now going to add those things together. So this plus that gives us our inspiratory capacity, the amount of air inspired in total. Likewise, our expiratory capacity is your normal breathing, so the amount expired through normal breathing, plus the amount of air expired through forcible exhalation. And we add those together, that's the total air expired. So our normal expiration plus the expiratory reserve volume gives us our expiratory capacity, the amount of air expired in total. So let's add it all together. We're going to take that tidal volume plus the inspiratory reserve volume plus the expiratory reserve volume. That is going to give us a number called the vital lung capacity, or VLC. So that's this arrow from the very top to the bottom of the ERV. And so that's going to be your total air moved in and out of your lungs per breath. Right? One big breath in, one big breath out. Now, finally, we can take all those amounts and add them together to get our total lung capacity. What can your lungs inhale and exhale? So this isn't going to include something that the vital lung capacity didn't include, and that's that residual volume. So remember, that's the amount of air that's still left in your lungs after you forcibly expired. So your total lung capacity, TLC, is going to be tidal volume plus your inspiratory reserve volume plus your expiratory reserve volume plus that last little residual volume. So basically the whole thing, total lung capacity, includes residual volume, expiratory reserve volume, tidal volume, and inspiratory reserve volume. So that whole thing gives you your total lung capacity. And we can translate this onto a graph just showing the actual volumes that are exchanged. So remember that our tidal volume is half a liter, so 500 milliliters. Our inspiratory reserve volume ranges. The max is about 3,100 milliliters. Our expiratory reserve volume ranges from 700 to 1,200. And our residual volume is about 1 liter. So when you add all those together, you can see that the vital lung capacity is going to range from about 3,100 milliliters up to about 4,800 milliliters. And your total lung capacity is going to range from about 4,200 milliliters on up to about 6,000 milliliters. So recall that females on average are going to be at the lower end and males on average are going to be at the higher end. Okay, so let's go over this graph one more time just to make sure everybody has it. This graph is important in respiratory physiology. It combines both pulmonary volume and pulmonary capacity on the same graph. So just remember, normal breathing, we're going to start right in the middle. This is our tidal volume, or TV. It's about 500 milliliters, half a liter. Our inspiratory reserve volume is the amount of air that you breathe in after normal breathing, and that's going to be these big peaks that start after the tidal volume, and they're going to be much higher, maybe around 3,100 milliliters. Your expiratory reserve volume down here, ERV, this is going to start at the bottom of the tidal volume graph and go down about 1,200 milliliters or so. And finally, we have our residual volume, RV, which is what's left in the lungs. It's about 1,000 milliliters or 1 liter. Now turning these into capacities, which means 2 or more of the volumes together, we have our inspiratory capacity, the amount of air exchanged both from normal breathing and from forced inspiration. That's tidal volume plus inspiratory reserve volume. And then we have our vital capacity, which is the total amount of air exchanged from normal breathing, forced inspiration, and forced expiration. And finally, we have our total lung capacity, which is our normal breathing, our tidal volume, plus our inspiratory reserve volume, plus our expiratory reserve volume, plus our residual volume left in the lungs. Hope you are able to understand this graph because, again, it's an important concept in respiratory physiology. So now that we understand pulmonary ventilation and we can quantify it and graph it, we are ready to go molecular and look at how oxygen and carbon dioxide specifically are exchanged in the lungs and in the other tissues of the body. So we're going to do this by studying 2 different kinds of gas exchange in the respiratory system. Pulmonary gas exchange, which is the exchange of oxygen and carbon dioxide between the alveoli of the lungs and the pulmonary capillaries that contain blood. We're also going to look at tissue gas exchange, which would be all the tissues of the body and how they're exchanging oxygen and carbon dioxide with the blood in the systemic capillaries. So the rule of thumb with this, to understand what's going on and how this oxygen and carbon dioxide move, is to understand the pressure gradients that we've been talking about before. So just remember, a pressure gradient is where molecules move from an area of higher pressure to an area of lower pressure. But each gas making up air has its own pressure, and that differs from the other gases that it's combined with. And so, in other words, these are partial pressures. So, for example, the pressure of oxygen is going to be different than the pressure of nitrogen, even though both are part of the air. The air is about 78% nitrogen, so it's going to have a different partial pressure than the oxygen does. This law of partial pressures is known as Dalton's law of partial pressures. And really all that's saying is that every gas is made up of things that different kinds of molecules, and each of those different kinds of molecules exerts its own pressure. And if you want to know what the total pressure of that gas mixture is, just add up the sum of its parts. So just add up the partial pressure of oxygen with the partial pressure of nitrogen with the partial pressure of argon plus the partial pressure of carbon dioxide, and then the pressures of all the other things that might be in the air. So this little graphic here kind of illustrates what we're talking about. So the total pressure of whatever gas mixture we're talking about here is going to be the sum of the individual partial pressures of the gases that make it up. So gas 1 pressure plus the pressure of gas 2 would make up the total pressure if the mixture was made of just those two gases. So actually a pretty simple concept, but an important one for understanding how oxygen and carbon dioxide are going to move in the system. Now, another law comes into play that's going to be important for predicting and understanding the behavior of carbon dioxide and oxygen across that blood barrier. And this is called Henry's law of solubility of gases. So what this is saying that the different gases that make up a gas mixture, in this case air, is going to differ in terms of solubility of each gas. So, for example, carbon dioxide is more soluble in water than oxygen is. This is going to play an important role, in addition to the role the partial pressures have, in determining where things are going to move. So different gases dissolve differently in water, or in the case that we're talking about here, we're really talking about plasma of blood, which is mostly water. So rule of thumb here, just remember as we go on talking about this, that oxygen solubility is less than carbon dioxide solubility in plasma or in water. So we're first going to start with pulmonary gas exchange, which again is the exchange of oxygen and carbon dioxide between the alveoli of the lungs and the blood in the pulmonary capillaries. So let's first start talking about oxygen. And so the movement of oxygen from the alveolus into the venous blood, how is that going to happen? Well, remember, things are driven by pressure gradients. So the pressure of oxygen in the alveolus is 104 millimeters of mercury. So that's a high pressure. So oxygen's under high pressure in the alveolus. We've inhaled that oxygen during inspiration, so it's come from the air outside. Now it's in our alveoli and it's under high pressure. It's rushed in there. So it's 104 millimeters of mercury, lots of oxygen inside of that alveolus. Whereas the pressure of oxygen in the venous blood is only 40 millimeters of mercury. So that's a huge difference. 104 in the alveolus, 40 in the venous blood. And so that's a strong pressure gradient, and that's going to allow that oxygen to flood in to the venous blood there, because it wants to go from an area of high pressure to an area of low pressure, right? It's a large pressure gradient. And so it's going to flood from 104 in the alveolus. It's going to go into the blood where it was only 40 before, and it's going to keep doing that until those pressures equalize. So that allows the oxygen that we breathe in to get into the blood system very fast and then be taken into the systemic system, where it will bring oxygen to our cells of our tissues so that it can undergo cellular respiration and they can produce the ATP our body needs. Now, the movement of carbon dioxide, remember, it's going to go basically in the opposite direction. You do breathe a little carbon dioxide in when you inspire, but most of it is going to go out during expiration. So if we look at the movement of carbon dioxide in the capillaries, remember that slightly there's going to be a lower partial pressure of carbon dioxide in the alveoli than there is in the blood. So in the alveoli, the carbon dioxide pressure is about 40 millimeters of mercury, whereas in the venous blood, it's about 45, 46. So there is a pressure gradient there, but it's a small one compared to what we have for oxygen. So 46, so it's a little bit higher in the blood than the pressure is inside the alveolus. So the carbon dioxide naturally wants to move out of the blood and into the alveolus. However, why is it still able to happen fast, given that that pressure gradient isn't as big as it was for oxygen? Well, let's not forget about Henry's law. Henry's law says that gases have different solubilities in water. And in the case of carbon dioxide, remember that carbon dioxide is more soluble in water, more soluble in plasma than oxygen is. So because of that high solubility in plasma, it can flow fast despite having not a large pressure gradient. And so the carbon dioxide with that small pressure gradient and with its high solubility is going to flow very quickly into the alveoli from the blood so that that carbon dioxide can be exhaled out. Just like there were several factors that could affect pulmonary ventilation, there are 3 factors that can affect pulmonary gas exchange. Number 1, the pulmonary gas exchange is going to be affected by the thickness of the respiratory membrane. 2, pulmonary gas exchange is going to be influenced by the surface area of the respiratory membrane. And 3, pulmonary gas exchange is going to be affected by how we get a matching between the flow of air and the flow of blood. So let's take a look at these, and you'll see here a scanning electron micrograph just showing alveoli and how that surface area of air exchange is increased by all those alveoli, as well as gas exchange from all these branches of these capillaries. So starting off with number 1, the thickness of the respiratory membrane. So recall that the respiratory membrane is the interface between the capillaries and the alveoli. And that thickness can increase during times of inflammation. So, for example, some kind of inflammatory response will make things swell. And so that increases the thickness of the respiratory membrane. And when that happens, you've increased the distance that gases like oxygen and carbon dioxide have to travel to be able to get where they need to go. So that extra time it's going to take for those gases to diffuse because of that inflamed membrane, that's going to decrease the efficiency of getting oxygen and carbon dioxide where they need to go. So decreases the efficiency of pulmonary gas exchange. 2, the surface area is very important. So again, the reason why you have all these branches and bubbles is you're increasing the surface area, and that gives more binding sites for erythrocytes, red blood cells, to come and be able to come in contact with the alveoli so that those gases like oxygen and carbon dioxide can be exchanged. Now, the third factor affecting pulmonary gas exchange is a phenomenon known as ventilation-perfusion coupling, or ventilation-perfusion matching. We're going to refer to ventilation with a V and perfusion with a Q. Now, this is the matching between alveolar airflow and capillary blood flow. So, in other words, ventilation is the air we take from the outside and bring down into our alveoli. And of course, blood flow, we're talking about pulmonary capillary blood flow. Now, what we want is for the airflow coming into the alveoli to be matched with the blood flow flowing through the pulmonary arterioles and pulmonary capillaries. We want that ratio to be 1-to-1, or in other words, about 1.0. Although in reality, the normal range is about 0.8 to 0.9. But we want that to be matched because think about it. If we had air coming in with the oxygen in our alveoli and there was no blood flow or not an adequate blood flow to butt up against the alveolar respiratory membrane, we couldn't have efficient gas exchange of oxygen. And by reverse, if there wasn't adequate alveolar airflow to match the blood flow bringing carbon dioxide back, we wouldn't be able to efficiently get carbon dioxide across the respiratory membrane and into the alveoli to be exhaled. So we want the airflow and the blood flow to match each other. So 2 rules of thumb here. 1, blood flow is directed to areas with the greatest airflow. And in contrast, airflow is directed to the areas of the greatest blood flow. So if you remember that, and remember that that needs to be the case so that we can get the gases across the respiratory membrane, this ventilation perfusion phenomenon is not so hard to understand. So let's look at this idea that blood flow is directed to areas with the greatest airflow. In other words, the higher the ventilation, so the deeper we're breathing in and bringing oxygen to our alveoli, as we know, that means that we're going to have the highest pressure of oxygen in the alveoli. So the more that comes in, we're filling up the alveolar volume, and thus the pressure is increasing. So we're having high pressure of oxygen in the alveoli. Well, that is going to have an effect on these pulmonary arterioles, these pulmonary capillaries. In fact, what's going to happen is those pulmonary arterioles are going to dilate. They're going to open up, and that allows for greater gas exchange to get the oxygen from the alveoli across the respiratory membrane and into those pulmonary capillaries and arterioles. So one way to look at it is if there's a lot of oxygen in that alveolus, the blood goes, oh boy, we better meet it there so that we can pull some of that oxygen out of the alveoli and get it to the tissues where it needs to go. Likewise, if we're not breathing in a lot, we're not taking in a lot of oxygen, and so our ventilation is low, then we're going to have lower pressure of oxygen in the alveoli. And so we don't need as much blood right there to exchange the gas. And so the pulmonary arterioles are going to constrict. So they're going to say, okay, there's not a lot of oxygen there, so let's not really try to bring any into the blood. Let's let it stay in the lungs. And so by constricting, you are going to be sort of blocking some of that oxygen from getting into the blood. The blood's not going to be going as fast to meet up with that oxygen. Okay, so let's look in reverse here, and that is that airflow is directed to the areas with the greatest blood flow. So in other words, let's say that your pulmonary capillaries were bringing in carbon dioxide, a lot of it, and so it had high pressure of carbon dioxide in the capillaries. This is going to cause the bronchioles in the lungs to dilate. In other words, there's a lot of carbon dioxide in the capillaries. Let's dilate and get that carbon dioxide into the lungs. Likewise, if the pressure of carbon dioxide in the pulmonary capillaries is low, because there's not as much carbon dioxide, well, now those bronchioles are going to constrict, which is going to limit the efficiency of gas exchange of carbon dioxide. So it's going to block a lot of that carbon dioxide from getting in. Now, what happens when we have a mismatch, when ventilation and perfusion rates are not the same? Well, we're obviously going to be far from 1.0, and this can happen from disease such as pneumonia or asthma, causing changes to the amount of ventilation that can get through. So let's say that there was blood flow happening at the lung, but not adequate ventilation, so low oxygen, for example. In that case, perfusion of the lung units is without the ventilation, and there's plenty of blood to take stuff away, but no stuff to actually take away. And so we end up getting unoxygenated blood entering the systemic circulation and not being able to do anything for our tissues. There's no oxygen for it to deliver to the tissues who need it. So that is called a shunt. And that's when the ratio of V to Q is 0. This is an extreme example. The opposite of that would be, okay, we have ventilation where we have all this oxygen in our alveoli, but there's not adequate blood flow to be able to meet up with those alveoli to take up the oxygen. In this case, gas enters and leaves the lungs, but there's no blood to carry it to the tissues. So it's a waste. It's wasted ventilation in a sense. You got the goods, but no transportation to take the goods away. So in that case, our VQ ratio is infinite, and we call that dead space, where there's gas, but no blood to take that gas anywhere. All right, and so now that we've talked about pulmonary gas exchange, let's talk about gas exchange in the tissues of the body. So tissue gas exchange is gas exchange between the systemic blood and the systemic tissues. Now recall that in order for cells to undergo aerobic cellular respiration, they have to have oxygen. And the whole point of aerobic cellular respiration is to make the energy currency molecule ATP. So they have to have oxygen to do it. But in the process, they're going to give off the waste product, carbon dioxide. So as they're taking in oxygen, they're building up carbon dioxide, and we need that carbon dioxide to leave the tissues and go into the capillaries. So how is that going to happen? So let's just look at oxygen first. So the movement of oxygen from the capillaries, where it was traveled to because of the lungs unloading the oxygen into the blood, and how we're going to get that oxygen into the tissues. So this comes back to those pressure gradients as well as solubility. So remember that because we've now taken that oxygen from the lungs, traveled it to the systemic capillaries, the partial pressure of oxygen in those systemic capillaries is now high. It's 100 millimeters of mercury. Whereas initially the oxygen's partial pressure in the tissues, these are tissues that are starved of oxygen, they need it to do cellular respiration, that pressure is going to be low at about 40 millimeters of mercury. So once again, a strong pressure gradient, except this time the pressure is highest in the capillaries for oxygen and lowest in the tissues. So which way does the oxygen want to go? Of course, out of the capillaries, down its pressure gradient into the tissues so that the cells can undergo aerobic cellular respiration. So, and it's such a strong pressure gradient that that oxygen is going to flow fast from the capillaries into the tissues. Good thing, because when we need oxygen, we need oxygen. It better be able to get in there fast. Carbon dioxide is going to go a little bit differently. So remember, there's less of a pressure gradient for carbon dioxide compared to oxygen. At least that's the way it was for pulmonary gas exchange. Turns out it's the same for tissue gas exchange. So in the systemic capillaries, the partial pressure of carbon dioxide is going to be rather low, about 40 millimeters of mercury, whereas it's going to be high in the tissues initially, about 45 millimeters of mercury, because all that carbon dioxide is being given off due to aerobic cellular respiration. So we do have a pressure gradient here, but it's slight compared to what it was for oxygen. It's going to be 45 in the tissues and 40 in the capillaries. So the carbon dioxide, just from the pressure gradient, is going to want to go from the tissues into the capillaries. But remember, Henry's law, carbon dioxide is more soluble in plasma and water than is oxygen. So even though the pressure gradient is small, it's still going to move fast from the tissues to the capillaries because it's highly soluble in plasma. So that's basically tissue gas exchange, same kind of phenomenon as we had for pulmonary gas exchange, except now we're talking about tissues and capillaries, and you got to keep in mind what's going on with aerobic cellular respiration. So by now you're probably familiar with the fact that there are many things that can affect how the respiratory system works. We talked about factors that could affect pulmonary ventilation. We talked about factors that could affect pulmonary gas exchange. Likewise, there are 3 factors that affect tissue gas exchange efficiency. One is kind of like we had in pulmonary gas exchange, and that is surface area. Except with pulmonary gas exchange, we were talking about the surface area of the alveoli in addition to the surface area of the pulmonary capillaries. With tissue gas exchange, we're specifically talking about the surface area of the systemic capillaries. We want as much surface available as possible to allow for oxygen and carbon dioxide to penetrate the tissues of the body. So the greater the surface area of the capillaries, the greater the efficiency of gas exchange in the tissues will be. And this is accomplished with the branching of the systemic capillaries. They branch and branch and branch all over the place, thereby increasing the surface area for gas exchange with the tissues. Additionally, the distance for diffusion. So a lot of the epithelial layers in our body are stratified. And if you recall, stratified epithelium means that there are cells in layers on top of each other. For example, in thick skin, you have many, many layers of stratified epithelium. Well, the larger that stratification is, the greater the distance is for gases like oxygen and carbon dioxide to diffuse across. And so the more distance those gases have to go to diffuse, the less efficient that gas exchange is going to be. And finally, like it was in the pulmonary gas exchange, the perfusion of tissue. So again, having blood flow going near where you need the gases to go is vital. And so when you have low blood flow to the tissues, you're going to have less gas exchange. It's not going to be as efficient because there's not as much area for the blood to pick up those gases. And if there's no blood, there's not going to be any gas exchange. So for example, this could happen if you had, for example, I don't know, something really tight on your arm, knocking off the blood flow to those tissues. Eventually, if that goes on long enough, those tissues are going to die because they're not able to get the gas exchange. So we want to make sure we are able to get oxygen, into the tissues and carbon dioxide out. Now let's talk about gas transport through blood. How do we actually get gases moving through the blood? Well, you might recall this protein, hemoglobin. So hemoglobin is the principal protein that's going to allow oxygen and carbon dioxide to travel through the blood. So let's talk about something called hemoglobin loading and unloading. In other words, how are we going to get oxygen onto hemoglobin and off of hemoglobin? So it's really important that we have the right amount of oxygen in our tissues. If you have too much oxygen, it's actually toxic. And if you have too little, our cells can't undergo aerobic cellular respiration to produce ATP. And once we can't produce ATP, it's game over. You can't survive without ATP. That's our energy currency that drives all the reactions in our body. So it turns out that most oxygen is going to travel through the blood loaded onto hemoglobin, and in fact, less than 1.5% of the oxygen in our body is going to be unbound. Now, each hemoglobin protein has 4 domains here, 4 chains, polypeptide chains. 2 are what we call beta chains, and 2 are what we call alpha chains. And in the middle of each of those polypeptide chains is what we call a heme group. And the heme group, right smack in the middle, has a single iron ion, so Fe2+. And it's the iron that the oxygen is going to actually bind with. So given that there's 4 irons in a hemoglobin, that means a single hemoglobin protein can hold up to 4 oxygen molecules. And when you add it all together, there's about 280 million hemoglobins per red blood cell in our body. Doing the math, that means that each red blood cell is going to hold over 1 billion oxygen molecules. So that kind of boggles the mind, but gives you an appreciation for the need our tissues have for that oxygen. So there's actually a reaction for loading oxygen onto hemoglobin, and that is when we have hemoglobin without oxygen, it's called deoxyhemoglobin. So HHB stands for hemoglobin. So hemoglobin plus oxygen then becomes the molecule oxyhemoglobin, HbO2. And so we can categorize hemoglobins as being partially saturated or fully saturated. So if only 1 to 3 of those heme groups have oxygen bound to them, we say that the hemoglobin is partially saturated. But if we fill all 4 sites with oxygen, then we say that the hemoglobin is fully saturated. Now, of course, there are things that can affect loading and unloading of hemoglobin and therefore affect gas transport through the blood. Now, recall that when you breathe in, the pressure of oxygen in your alveoli is going to be very high compared to the pressure of oxygen in the venous capillaries, in the pulmonary capillaries. And that created a large pressure gradient, and that oxygen wanted to flood into those capillaries from the lungs. Well, now it's all in the capillaries. And so by the time it's in the systemic circuit, there's a lot of oxygen there. And so the pressure of oxygen is going to be very high in the capillaries compared to the pressure of oxygen in the tissues, where the tissues are using up oxygen as fast as they get it for cellular respiration. So because of that, the capillaries in the systemic circuit are going to have a lot of oxygen available to be loaded onto the hemoglobin. And in fact, about 97 to 100% of the hemoglobin molecules in the systemic blood are going to be fully saturated with oxygen. They're going to have all 4 oxygens that they need. Well, when this happens, these hemoglobins get greedy and they're like, okay, I got all my oxygen and I'm not letting it go. So they have a very high affinity for their oxygens. When there's 4 oxygens, when they're fully saturated, they do not want to let that oxygen go. And it's going to take a really high pressure gradient between the tissues and those systemic capillaries to get the hemoglobin to release oxygen. So when that pressure gradient is high, which it is, because remember, it's about 100 millimeters of mercury in the systemic capillaries compared to about 40 millimeters of mercury in the tissue. So there's a high pressure gradient, really strong pressure gradient. That oxygen is going to forcefully want to move from the systemic capillaries where the pressure's high to the tissues where the pressure's low. Now, but again, the hemoglobin wants to hang on to it. So when that pressure gradient is high enough, it will finally release one of those oxygens. And now you have 3 oxygens bound to that hemoglobin. But it's still kind of greedy. It doesn't really want to give it away, but it's going to give them away now easier than it did before. When it was fully saturated, it really didn't want to give them away. But with a strong pressure gradient, okay, now it's going to give one away. And now the more it gives away, the more it's going to give away. The affinity of the hemoglobin for the oxygen is going to decrease. So after 40 millimeters of mercury of oxygen in the blood, the hemoglobin starts giving it up, because what's going to happen is when the affinity changes and due to the pressure gradients, that hemoglobin 3-dimensional protein is going to have conformational change of shape. It's going to change its shape, and that affects the ability of that hemoglobin to hang on to the oxygen. And so the more you lose, the more you lose. And by the time we're down to 25 millimeters of mercury, the hemoglobin only has one oxygen left, and it's ready to get rid of it and be fully unloaded because it no longer has that affinity. It's changed shape so much that it's giving away its oxygen like candy at that point. So the lower the pressure of oxygen in the venous blood, the more that it'll unload the oxygen into the tissues where it's needed. So this phenomenon here, this graph, is called the oxygen hemoglobin dissociation curve, and it's correlated with the percent of hemoglobin saturation that we have. Now, there are a lot of laws in physiology that are named after different people. So we've already talked about Dalton's law and Henry's law. Well, here's another one for you. This is called the Bohr Effect, which tries to explain some other factors that can affect the ability of hemoglobin to unload its oxygen. So the Bohr Effect says that hemoglobin will decrease its affinity for oxygen under certain conditions because of that unfolding, that denaturing, or the conformational change that occurs in that three-dimensional protein. So remember, as it changes its conformation, it's going to have less affinity. It's going to more easily give up those oxygens. And principally, this can happen with pH. So as pH decreases, as it becomes more acidic, if you remember back from some of your early lessons you learned about proteins, you'll remember that proteins denature in acidic environments, under high temperature, under high electrical gradients. And so the Bohr effect specifically addresses pH. The lower the pH, the more acidic the pH, the more conformational change of shape of that hemoglobin you're going to have, and therefore it's going to start giving up its oxygen. But high temperatures, just like with any protein, cause more unfolding. And if we have high carbon dioxide pressure in the tissues, that's also going to affect the ability of hemoglobin to retain its oxygen. There's another compound that's going to affect this. This is called BPG, and it's something that our red blood cells will give off as they're undergoing glycolysis. And so the more BPG that occurs, the less affinity hemoglobin is going to have for its oxygen, and it's going to give up the oxygen more easily. It's going to have more unloading. So now let's talk about how we're going to get carbon dioxide out of the tissues and into the blood so that it can be taken back all the way to the lungs to be expired. So very little of carbon dioxide is going to travel directly in the plasma, and only about 20% is going to be bound to hemoglobin. So that's quite a bit different than what we were just talking about, where you know that the vast majority of oxygen molecules are going to be bound to hemoglobin on the iron ion that is in the heme groups of the hemoglobin. Now, the 20% of carbon dioxide that is bound to hemoglobin is not going to be bound to the iron ions. It's going to be bound to polypeptide chains in the red blood cells and thus form a compound called carbaminohemoglobin. However, 70%, so the vast majority of carbon dioxide molecules leaving the tissues, are going to be transported through the blood in something called bicarbonate, in a form called bicarbonate, or HCO3-. So it's a negatively charged ion. So again, carbon dioxide is produced as a waste product of aerobic cellular respiration in the tissues, and those tissues have to get that carbon dioxide out. So thanks to the pressure gradient and the solubility of carbon dioxide in water, we have carbon dioxide leaving the tissues, going into the blood plasma, while the oxygen's entering the tissues from the red blood cells. So very little is dissolved in the plasma, a little bit is. 20% will bind to the hemoglobin in the red blood cells, and the rest is going to form bicarbonate. So let's see how that happens. So we have the carbon dioxide leaving the tissues, meeting up with these red blood cells, and as soon as they do, as soon as they get in those erythrocytes, they are going to meet an enzyme called carbonic anhydrase. Remember, anything ending with an ASE is an enzyme. So carbonic anhydrase. And like so many other enzymes, this enzyme is going to serve as a catalyst to speed up the reaction of carbon dioxide and water to produce the compound H2CO3, which is carbonic acid. So carbonic anhydrase is an enzyme that catalyzes the reaction of carbon dioxide and water to form carbonic acid, H2CO3. Now, it's not going to stay carbonic acid very long. It very quickly dissociates into bicarbonate ion, HCO3-, and free hydrogen ion. So that H2 right here, one of them is going to become part of bicarbonate. The other one is a free positively charged hydrogen ion. Now, you'll remember that when hydrogen ions are given off, that by definition is how you produce an acid. So the higher the concentration of hydrogen ions in a solution, the more acidic the solution is. Now, a lot of those hydrogen ions are going to bind to the hemoglobin, again, not with the iron ions, but in other parts of the hemoglobin. But the bicarbonate is actually going to enter directly into the plasma. Now, given that bicarbonate is a negatively charged ion, we'd be in some trouble if it just went out of the red blood cells into the plasma and nothing else happened, because we'd be adding all these negative charges there and all these hydrogen ions, and that would acidify our blood. So we have to have something that will balance that. And the way we're going to balance it is that chloride ions, Cl-, are going to go from the plasma into the red blood cells. So they're coming in while the bicarbonate ions are going out. So it's kind of balancing that negative charge that we would be putting out if it was only the bicarbonate. So it's going to kind of neutralize that. That process of the chloride ions coming into the red blood cells while the bicarbonate goes out, that is called a chloride shift. And that chloride shift of shifting chloride ions into the red blood cell while the bicarbonate ions go out is going to help to maintain the correct pH in the blood plasma. Now, let's talk about what's, in a sense, the reverse of what we just saw of how to get carbon dioxide out of tissues into blood. Now let's talk about how to get the carbon dioxide from the pulmonary capillaries, from the blood and the pulmonary capillaries into the alveoli of the lungs. So it's basically the reverse reaction. We already have bicarbonate in the plasma now because that was done during the tissue gas exchange. And we have chloride ions already in the red blood cells from the chloride shift. So now what's going to happen when we come near the membrane, the respiratory membrane of the alveoli? So here's your alveolus. Here's your inside your pulmonary capillaries, and here's a red blood cell. The bicarbonate is going to enter the red blood cell while the chloride ion goes back out. And now this bicarbonate is going to do the reverse reaction of what we saw before. So we have our bicarbonate. It's going to meet up with the hydrogen ions that are bound to the red blood cells. That forms carbonic acid, as you know. And now that carbonic acid is going to dissociate with the help of the enzyme carbonic anhydrase into carbon dioxide and water. And at this point, now we're back to pressure gradients and solubility rules. And that is going to bring the carbon dioxide back into the alveoli. And as we know, oxygen's going the other way. So now that carbon dioxide is going to build up in the alveoli, and eventually out it goes when we expire. So this concludes our lecture over respiratory system physiology. You've learned about pulmonary ventilation, pulmonary and tissue gas exchange, gas transport through the blood, and unloading and loading of hemoglobin. I hope you've enjoyed it, and I will see you next time.